Yeah, Dhanush, once again you can give the explanation on deproteination of blood.
When the protein present in the blood precipitated by tungsten acid, the filtrate is known as protein-free filtrate. This method is based on the precipitation of protein in blood by large ion. On adding the acid, the blood is lagged very rapidly and the hemoglobin is changed to acidic remitting. No precipitate is obtained until the sodium tungstate is added. The color is residue is reddish brown and the color of filtrate is colorless.
Okay, thank you. Thank you, Dhanush. Is there anybody willing to share points on deprotonating method of blood? Others also can give answers. Saundarya, Harish, Sudarshan. You also can share the points regarding deprotonation of blood. What are the different deprotonating agents available for making blood gets eliminated with proteins? And what are the blood types of blood samples will be analyzing? What kind of blood samples will be analyzing? Can anyone answer? Even you can post your answers in the chat box, since the messages also are recorded. Your answers will be recorded. And it will indicate whether you are active during the class hour. Whenever we ask question, you should respond, okay? So no response. What are the types of blood samples we'll be analyzing? Yeah, plasma, whole blood sample, and another one. What are the three samples will be analyzed? Yeah, serum. Thank you, Harish. And Subhikshna. Surya Prakash also has given answer. Pramod, Akash, okay, very good. Serum samples. Serum, plasma, and whole blood will be analyzed. So we are going to analyze either whole blood or plasma or serum. You know the difference between serum and plasma, right? What is the main difference between serum and plasma? In serum, we'll be preparing clotted blood. Supernatant liquid of clotted blood is called serum, where plasma will be added with deprotonating agents, right? My voice is breaking, huh? Just wait. Since I'm in another meeting also, that's why. Now is it clear? Whether my voice is clear now? Yeah, plasma contains protein, so it will be deprotonated. Whether my voice is clear now? So what is the difference between serum and plasma? We need to add anti-clotting agents, right? Anti-clotting agents to plasma sample. That's what anticoagulant we need to add. Where with serum, clotted blood is supernatant liquid of clotted blood is taken. Where we will not add any reagent. Sometimes to improve the clot formation, we'll add some reagents, or else we'll allow to make the clotted blood, then the supernatant liquid is taken. And here anticoagulant is added in order to avoid the formation of clot. And once all the cells have been precipitated in the supernatant liquid will be having plasma, where we can analyze plasma proteins that is present in the plasma. and whole blood directly would be taken, and that also will be stored with anticoagulant in order to avoid clot formation. And one more sample is with deproteination. We'll be removing proteins present in plasma by means of deprotonating agents like tungstic acid and trichloroacetic acid. Okay, now let's move on to the next experiment. I hope you can see the document shared for the next experiment. That is eighth experiment, quantitative estimation of serum cholesterol by Liebermann-Burchard'S method. Okay, quantitative estimation. So we are going to analyze serum cholesterol level quantitatively by using Liebermann-Burchard'S method. Okay, whether you are getting my voice clearly.
Yes, ma'am.
Okay, thank you. Okay, then we'll proceed. Then aim is to determine the amount of cholesterol present in the given sample of human serum. Here, serum sample is taken by Liebermann-Burchard'S method. Amount of cholesterol is going to be determined by using this reagent, Liebermann-Burchard'S reagent method. Okay, so what are the chemicals we need to use? Means standard cholesterol solution. Standard means known concentration of cholesterol solution is prepared. Okay, according to the procedure given, the mentioned concentration of cholesterol solution is taken. That is called as standard cholesterol solution. Standard represents known concentration, and the reading will be compared with a sample. So it will be the reference standard solution. Then next one, stable Liebermann-Burchard'S reagent. Just a second. Stable Liebermann-Burchard'S reagent, then ice cold acetic anhydride, ice cold condition acetic anhydride. That is the reagent taken to prepare Liebermann-Burchard'S reagent. Then glacial acetic acid, here solvent used for preparing cholesterol solutions, both standard and sample solutions with glacial acetic acid only. Since cholesterol is fat, it will be dissolved in glacial acetic acid. And then ice cold concentrated sulfuric acid. This will be of concentrated and will be fuming, so that we have to use ice cold concentrated sulfuric acid. So these are all the chemicals we require for Liebermann-Burchard'S method. Okay, standard cholesterol solution, stable Liebermann-Burchard'S reagent, and even we can prepare manually Liebermann-Burchard'S reagent. It is available ready-made reagent also, and we can prepare with reagents like acetic anhydride, acetic acid, and concentrated sulfuric acid. So for that purpose, we require these chemicals. Then apparatus required are calorimeter. Already I said calorimeter or visible spectrophotometer with electromagnetic radiation, visible radiation UV, visible radiation will be applicable to the sample where cholesterol is present to analyze the how many absorbing species cholesterol molecules are existed in the sample. Okay, since absorbance is directly proportional to concentration, what parameter in calorimeter will be analyzing means absorbance, and that will be proportional to the concentration of cholesterol present in the solution. Okay, that's why apparatus calorimeter is required. Then glass sample cell or cuvette, it will be called as cuvette. Let me write the spelling for cuvette in chat box. Okay, cuvette. That is called as sample cell. Even glass sample cell may be called as cuvettes. Okay, so where we will be taking the prepared samples like standard, sample, blank, and all will be taken in the sample cell, and it will be placed in the calorimeter to analyze the sample. Okay, and incubator at 37 degrees Celsius. In order to prepare solutions and make it stabilize, we need incubator at 37 degrees Celsius. Then test tubes. test tube stands. Test tubes are required to prepare the samples, various samples, standard, sample and test. Three kind of sample will be preparing, for that will be prepared with the test tubes. So we require test tubes. Then test tube stands to hold the test tubes. Then pipettes to take the solutions in order to prepare standard and samples. In order to add all these measured reagents, we need pipette with the 10 ml capacity and 1 ml capacity. Then tissue papers in order to clean the glass sample cell, that is cuvette, sample cell of calorimeter will be cleaned by means of tissue paper. So these are all the requirements, accessories and apparatus required for quantitative estimation of serum cholesterol. What are the chemicals we required? Cholesterol solution, sample cholesterol substances also we require, then stable Liebermann-Burchard'S reagent, ice cold acetic anhydride, glacial acetic acid and ice cold concentrated sulfuric acid. Apparatus required, a calorimeter, instrument calorimeter, along with the glass sample cell and tissue paper for that. Then incubator at 37 degrees Celsius to maintain the standards at this particular temperature, 37 degrees Celsius. Then test tube stands, pipettes. These are all the requirements. Let's move on to the principle. Serum is mixed with stable Liebermann-Burchard'S reagent. So this is the reagent and serum will be having cholesterol. Acetic acid dissolves cholesterol, which later undergoes Liebermann-Burchard'S reaction. So which dissolves cholesterol present in serum? Acetic acid. So acetic acid will be the solvent for sample and for standard, mainly for cholesterol. It will be dissolving cholesterol. Then it undergoes Liebermann-Burchard'S reaction. Normal serum cholesterol level lies within the range of 100 to 240 milligram per 100 ml or 100 to 240 milligram per deciliter. Also 100 ml represents milliliter, represents deciliter. Then in healthy adults, this is the normal range of cholesterol, and already cholesterol normal range for women, men, everybody we have discussed in the theory itself in the topic of lipid metabolism. Kindly go through that. Okay, then average serum cholesterol level is about 200 milligram per 100 ml, that is milliliter, at 25 in men, and rises slowly with the age of reaching a peak figure at the age of 40 to 50. Okay, so it will be rising slowly at the from 25 to 50. When it is increasing with the age, cholesterol level also will be slowly increasing. Then it will be declining. Okay, then serum cholesterol of 300 milligram per deciliter or more in young adults, which causes a serious indication of coronary disease. So 300 milligram is the indication of coronary disease. Coronary means heart diseases. Okay. Serum cholesterol increases in many pathological conditions like lipemia, leprosis, diabetes mellitus, hypothyroidism, biliary obstruction, hepatitis, and prolonged atherosclerosis. These are all the lipid metabolic disorders. Already we have discussed regarding pathological conditions of cholesterol. You can quote the same disorders with description here, along with other diseases also. You know what is diabetes mellitus, and then atherosclerosis. This all we have already discussed, and related to that disorders are lipemia, leprosis, hypothyroidism. This all. Okay. Decreased serum cholesterol level is found in hyperthyroidism, pernicious anemia, and hepatocellular damage and acute infections. So these are all pathological indications, clinical significance of cholesterol. Okay. Next, we'll move on to the procedure. I'll be giving you how the reaction takes place between Liebermann-Burchard'S reagent and sample and your sample. Okay. Now let's discuss about the procedure. Meanwhile, I'll send you the video of Liebermann-Burchard'S reaction so that you will understand the concept behind it. Okay. Just wait. I will be sharing with you that reaction so that you will understand the live video. If I keep, you will understand better about Liebermann-Burchard'S test. Anil, let me share with you in the classroom. Okay, the video will be shared with you in classroom. How the Liebermann Burchard's reagent reacts with the fat cholesterol. Okay. Now I'll describe about the procedure. So you need 3 test tubes in order to prepare blank, standard and test. You can observe the side headings: blank, standard and test. Okay. So we require 3 kind of test tubes. In the blank test tube, 3 test tube you have taken to prepare blank, standard and test. Blank means which contains no sample, where in standard known concentration of sample will be added, and in test unknown sample is added, unknown concentration of sample is taken. Okay, so this standard and test will be having active ingredient, where blank will not be having an active ingredient, that means sample or analyte, whatever biomolecule, whatever you need to analyze, where it will be having only reagents added to sample. Okay, so that is called blank. Why we have to prepare blank means the reagents also will have absorbance in calorimeter. Since we are going to analyze the samples by using calorimeter, so it will be absorbing in calorimetric region. So that blank need to be prepared with only reagents, except sample, whatever reagent we take, that is to be prepared as blank. Okay, so whatever absorbance is absorbed by the reagents like glacial acetic acid and Liebermann Burchard's reagents will be eliminated from the analysis of sample. For that blank is prepared. Okay, so how you will be preparing means in all 3 test tubes, we have to take here first you take 0.1 ml. First quantity is 0.1 ml. See, in all the test tubes you are going to add 0.1 ml quantity of something. Let's see what you are going to add. 0.1 ml of glacial acetic acid is added to blank. Then standard also 0.1 ml of standard cholesterol solution is taken. Standard is known concentration solution is taken. Then take the standard preparation. I will be giving you in the reagents preparation column. So you will be knowing how to prepare standard cholesterol solution. So from that 0.1 ml standard cholesterol solution is taken. Then test tubes you have to add 0.1 ml serum. Serum is your unknown concentration of cholesterol, right? Whatever cholesterol present in serum is unknown, you are going to determine. Where in standard you know how much concentration of cholesterol you have taken of 0.1 ml. So all the tubes you have added 0.1 ml, where blank only added with glacial acetic acid. Since glacial acetic acid is solvent for cholesterol, we are adding only solvent for cholesterol. Where a standard solution is added with cholesterol solution. Solution represents cholesterol has been dissolved in glacial acetic acid, right? So except cholesterol, we need to take the solvents added in blank. So blank is what? 0.1 ml glacial acetic acid. Standard is what? 0.1 ml standard cholesterol solution, and test is 0.1 ml of serum. Next, what we have to add? Only 6 ml of stable Liebermann Burchard's reagent. We need to add 6 ml to all the test tube of reagent. Same quantity we need to add. So that is only the preparation of your sample test tubes. Okay. So what you are going to take? 0.1 ml sample first. Then you are adding 6 ml stable Liebermann Burchard's reagent to all 3 test tubes. Okay. So that is what the preparation procedure. Then mix well, all the solutions to be mixed, not together in each test tube. It must be mixed with particular sample. And then incubate all the tubes at 37 degrees Celsius for 18 minutes. 18 minutes we need to incubate at 37 degrees Celsius in the incubator. And so that meanwhile in the 18 minutes, within 18 minutes, the reaction will be completed between cholesterol and and Liebermann-Burchard'S reagent. Okay, and set the photometer or colorimeter to 0 optical intensity or absorbance with blank. This 0 optical density or absorbance is made with blank. Blank absorbance should be 0. Then only the that means absorbance of reagents will be 0. Then we have to read the optical density. Optical density or absorbance, it is called as optical represents radiation density. That means amount of radiation absorbed by cholesterol molecules. So that can be said as absorbance. Let me write the means spelling of absorbance. Somebody will understand this term like absorption. It is absorbance. Okay, optical density or absorbance will be measured. Then optical density will be read for the standard and tested 620 nanometers. 620 is colorimetric region. Colorimetric region of electromagnetic radiation will vary from 400 to 800 nanometer. Okay, 400 to 800 nanometer. So this wavelength, this is wavelength. 620 nanometer represents wavelength. Wave represents radiation. What radiation? Visible radiation from electromagnetic spectrum. Visible radiation by means of colorimeter is sent to the sample at the wavelength of 620 nanometer. Nanometer is unit for wavelength. Okay, distance unit. Wavelength is length representing distance, so that the unit is 620 nanometer. Nanometer unit. Okay, 10 to the power of minus 9 meters. Okay. 620 range is 620. So at this wavelength, all the samples are tested for its optical density or absorbance in colorimeter. Okay, what is the colorimetric range? Visible radiation range. What is the range? I said 400 to 800 nanometer. Okay, in that range you will be getting, means you will be analyzing your sample from means your sample at 620 nanometers. 620 is lambda max or maximum absorbance for cholesterol at that particular wavelength. So that may be called as lambda max. Okay, 620 is lambda max for cholesterol. Got it? Then after analyzing, you need to write your readings in the tabular column. Now let me show you. In the another page, we can find how to prepare the reagents. Liebermann-Burchard'S reagent is prepared manually by adding, even we can get a ready-made sample, ready-made reagent also, but we could prepare with the available reagents. So what is the composition of Liebermann-Burchard'S reagent means? It composed of 220 ml of ice cooled acetic anhydride. Nowadays we cannot buy acetic anhydride from the chemical store, so instead of acetic anhydride we can add acetyl chloride for that. Okay, so acetic anhydride can be replaced with acetyl chloride, which is available since it is explosive in nature. Acetic anhydride is not sold nowadays. Okay, so in place of acetic anhydride we can find acetyl chloride. So 220 ml of acetyl chloride with 200 ml of glacial acetic acid is taken, and to that we need to add 30 ml of ice cold concentrated sulfuric acid, and it must be stored in a brown bottle, amber colored bottle at 4 degrees Celsius. So in freezing condition, it must be stored for further use. In order to increase the stability of Liebermann-Burchard'S reagent, it is stored in brown bottle at 4 degrees Celsius. So storage condition should be noted for this reagent. Okay, that is why it is called stable Liebermann-Burchard'S reagent. What are the reagent composition? Acetic anhydride, glacial acetic acid, concentrated sulfuric acid, and it is maintained at ice cold condition. So what will be the temperature? 4 degrees Celsius. Okay, what is the quantity of acetic anhydride? 220 ml. Where what is the quantity of glacial acetic acid? 200 ml. Concentrated sulfuric acid is 30 ml. Okay, so everything should be stored for further use at 4 degrees Celsius. Okay. So this is how the reagent is prepared. So after analyzing your the absorbance, you need to write the optical density at 620 nanometer. No, 620 or 520. I have given 520. It is 620. 620 nanometer. Wait, let me check with the another record. It is 620 or 520. It is 620 only. That is typographical error only. So 620 nanometer, where blank will be of zero optical density at that particular wavelength, since blank reagents are having zero absorbance. Once it is run with blank, then standard run we have to measure. Test run should be measured. Okay. From the old record, I'll give the reading now. Now you have to do only the calculation with the given formula. Okay, serum cholesterol, that is equal to optical density of test optical density divided by optical density of standard. This is direct comparison method. Test reading is directly compared with standard. Okay, so this is one of the quantitative method to find out the amount of biomolecules. So it is an analytical method. So be thorough about this method, direct comparison method. Mostly will be following this method only for deducting all the biomolecules when you are going with colorimetric analysis. Direct comparison method. And one more is calibration curve method. Here mostly you will be going with the direct comparison method. Direct comparison means test reading is directly compared with standard. Only one standard is taken. Where in calibration curve method we will be using multiple standards that will be discussed when calibration curve method is coming in the forthcoming practicals. Okay, so this is example of direct comparison method where optical density of test is directly compared with the standard, and 0.2 will be the concentration of standard. 0.2 is concentration of standard, and 1 will be this a dilution factor. Okay, and then 100 will be 100 ml. In order to get a percentage, 100 ml, that is milligram per 100 ml we need to take now. So that so the values representing that dilution factor and concentration of standard. Okay, so that we could get the concentration of unknown. Concentration of standard and concentration of means optical density of standard and test will give the reading of results for concentration of cholesterol present in the sample. Unknown concentration can be determined. So 0.2 divided by 1 represents dilution what you have made for the sample, and concentration of standard. This all will be the readings for 0.2 divided by 1. This is constant. For any sample if you take for cholesterol, that will be constant, 0.2 divided by 1. And 100, in order to get milligram, since normal reading how you are mentioning 200 milligram per 100 ml. So in 100 ml how much quantity of cholesterol is present? To determine that, 100 is added here. Okay, so you are multiplying with the given readings and you will get the amount of cholesterol present in the serum given sample. Serum is how it is prepared, clotted blood. So you will be collecting the blood, from that you will be preparing serum. That serum is taken for the analysis, and whatever result through that calculation you are getting, you need to fill up here. Okay, so now I'll give the reading. After then you need to do the calculation, and in the discussion what you will write, what is the actual application of this cholesterol procedure, cholesterol experiment. Once you are determining the normal level or level of cholesterol, it will indicate whether it is normal or abnormal condition cholesterol is present, and what are the diseases. Increased level, what diseases it will cause, and at decreased level, what diseases it will cause. What are the normal levels? Already I have given in the principle. Again, you write it under the discussion procedure. And what other methods are available to detect the cholesterol? If Liebermann Burchard'S reagent is not available, what other reagent methods are available to detect cholesterol? Other diagnostic procedures also to be included under discussion. So what all you will be writing under discussion? Application of the current method what we have discussed now, Liebermann Burchard'S method for determining cholesterol, and you need to find out what other methods and what are the clinical significance of this method, and what are the normal levels and abnormal levels of cholesterol, range of cholesterol. Okay, this all to be discussed. Now let me share with you the old record readings so that you can copy in your observation, write it and submit it for evaluation. Now you can find the PDF of previous record, that is 2015-16 batch record. It was handled by me only, so that I'm sharing with you the same experiment, quantitative estimation of serum cholesterol by Liebermann Burchard'S method. Is your 6th year seniors record. It is okay. So from them they have performed and they have got this reading, that is standard reading as 0.06 and test reading as 0.05. Note down this reading in your tabular column. The observation first noted down, and you have to apply this in this formula what we have seen already. 0.05 as test and 0.06 as standard, and these are constant readings, and this says about concentration and dilution of standard, and 100 represents in 100 ml. What is the quantity of cholesterol in 100 ml, milligram per 100 ml. In order to get milligram per 100 ml, it is to be added, multiplied with 100, and you will be getting around 166.66. And all of you don't put the same reading near to the standard level, that is normal level of cholesterol. You can alter the test reading. Keep standard as 0.06 only. All of you take 0.06 as standard reading, constant, and test reading only should be changed according to the normal level. Now she is getting 166, right? 166 milligram per deciliter in this record. The person who has done has got around 166 milligram per 100 ml, but the level varies between 100 and 200, right? So you can change the test results. You can increase or decrease accordingly to get the readings between the normal range. Then you put the results and submit it, okay, for evaluation. Include discussion part also and references. And this is what we have discussed in the last class. That is preparation of whole in tungstate filtrate. So already all have done. That is good. So the 8th experiment they have performed as 7th experiment. So you have got the result now. Okay. Now I'll post in YouTube video also. There you can find the demo of preparation on doing Liebermann Burchard's method. So just go through that video, YouTube video. It is explained by other college people, and you can gain knowledge regarding the color change and all. Now I have given explanation whatever you are going to do according to your manual, and it is referred from the practical manual only. Procedure is referred from practical manuals available. Then according to the PCE syllabus, in your syllabus this experiment is prescribed. So based on that it is designed to explain to you. Is there any doubt now? Have you all noted the readings? 0.06 is standard reading, where 0.05 is test reading. You can alter only test reading. Okay, you can alter only test reading, and you can calculate according to the accordingly to get normal values. Clear now? Is there any doubt? Again my voice is breaking up. Whether you got my explanation clearly?
Yes, ma'am.
Okay. Okay. Have you all noted down the readings? Now do the calculation, write it down, the chemical reaction between whatever you have written for previous experiment, chemical reaction and principle. exact principle, a chemical reaction between cholesterol and Liebermann Burchard'S reagent, what color it will indicate, and that color intensity is measured with calorimeter at 620 nanometer. This all principle you need to write it down, okay? And then do the calculation part, chemical reaction, mechanism behind analysis, quantitative estimation, and then you need to write the diagrammatic presentation, draw the diagrammatic presentation of color change between blank, test and standard, okay? So that that color intensity is measured with calorimeter at 620 nanometer. Here you can observe standard is giving 0.06, that means absorbance is high, where standard concentration level is high, and sample concentration is less. What, Harish?
No, ma'am.
Do you have any doubt?
No, ma'am.
Then keep quiet. And you can find the test is showing 0.05, so that test concentration is lesser than the standard. The absorbance is less, since absorbance is directly proportional to concentration. That is, the concept is taken to analyze the concentration of cholesterol present in serum sample. Got it? And you have to write discussion, as I have told. Whatever instruction I have given, accordingly prepare, write discussion along with references. Whatever net source you refer, give the link address for that, okay? And submit before 4 o'clock today. I'll assign the assignment to submit like previous class, how you have submitted. So the same way I'll be assigning now. Okay, thank you all. Is there any doubt?
Before 4, ma'am.
Yeah, take 4:30, up to 4:30 you can submit. I'll assign timing up to 4:30. Okay. So you'll be having getting 45 minutes time. Meanwhile, do the calculations, write mechanisms, discussion, everything, and draw the schematic representation of cholesterol estimation. Okay, Pa, thank you. Shall I stop presenting now?
Yes, ma'am.
Okay, then go with the calculation part. Thank you. Thank you all. Take care.
Thank you.
Okay, avoid all
Thank
misbehaviors.
you, ma'am.
Whoever is doing, just avoid all misbehaviors. Kindly cooperate with your staffs. Okay, whoever is coming to your class, just be obedient in class hours. You can show your all the things with others. Okay, and whatever you are showing misbehaviors, it will be again reflecting to you in some or other way in your future. Okay, so kindly take care of you in all the aspects. Okay, thank you all. Yeah.
ப்ரொசீஜர் எழுத வேணாம்ல மேம்?
ப்ரொசீஜர் வேண்டாம்ப்பா, நான் ஸ்கீமேட்டிக் ரெப்ரசன்டேஷன் தான் கேட்குறேன். இப்போ யூரின் அனாலிசிஸ்க்கெல்லாம் நீங்க ஸ்கீமேட்டிக் ரெப்ரசன்டேஷன் போட்டீங்க இல்லையா?
ஓகே மேம்.
அது மாதிரி போடுங்க. ஓகேவா?
ஓகே மேம்.
டெஸ்ட் டியூப்ஸ் 3 டெஸ்ட் டியூப்ஸ் ட்ரா பண்ணிட்டு 1 டெஸ்ட் பிளாங்க் ஸ்டாண்டர்ட் அப்படி போட்டுட்டு நீங்க என்ன ஆட் பண்ணீங்க, எவ்வளவு ரியேஜென்ட் ஆட் பண்ணீங்க அதெல்லாம் போட்டுட்டு என்ன கலர் சேஞ்ச் வரும், அந்த கலருக்கு நீங்க கலர்ல ஷேடிங் கொடுங்க. அதே மாதிரி இந்த டீப்ரோட்டனேஷன் மெத்தடுக்கும் நீங்க அப்சர்வேஷன்ல டயக்ராம் போட்டுக்கோங்க. என்ன கலர்ல டீப்ரோட்டனேஷன் ஆன பின்னாடி என்ன கலர் வரும், பிஃபோர் டீப்ரோட்டனேஷன் கலர் ஆஃப் த சாம்பிள் என்ன. ஓகேவா? அது ஸ்கீம் போட்டு வச்சிட்டீங்கன்னா டியூரிங் எக்ஸாம்ஸ் whenever you are means revising the procedures, you will be understanding better if you see the diagram itself. Okay, and it will be helpful to score more marks also in experiments, and you can easily remember to perform the experiments. You will understand the order of adding reagents also when you are putting the schematic representation. Okay, thank you all.